So I think today it will effectively mostly be um, my last uh, day of talking about plotting with ggplot. I think we'll revisit this again whenever we get our expression data analyzed. But for now, I think we're just going to this is the last kind of fundamental um, day that I'm going to spend on this. I don't think I'm going to spend that much time on it, but um, what I am going to do is kind of talk about a couple of things I missed on Wednesday, a couple of new things, and but I'm still going to be working with this graph, um, this plot that I made on Wednesday with our density plots. I put a couple of links in the description. These are one is about cowplot and how we can make multi-pane figures in one using cowplot, and the other is really just kind of a very um, nice summary of all the what we can actually do to a figure, of all the different variable things we can do um, with our figures to make them look really nice and really presentable, and also display information in a valuable way or a very easy to understand way. So today, let me get this script going again. Let's see what we're looking at. See what the figures we're looking at, just to remind ourselves of anything. Length. Okay, so let's see. At the inbox plot. So this is a box plot of our links. So this is a box plot of the same thing. We also have our density plot. Um, that's just a redrawing the same data, so it's a little bit easier to look at. So I did mention that if we're making a single ggplot figure, we can't use two different data frames and draw a single figure, right? So these are two different. I mean, it's a two different figures of the same data, but in many cases, especially uh, if we're presenting relatively similar data, we may want to present it side by side in the same graph, right, in the same overall figure. And so there are tools in R that let us do this. So one of the more kind of, I don't know if it's growing or it's popular already, but one of the more popular plotting packages, cowplot, and it just stands for COW plot. And it's Klaus O. Wilk. He is he's at U Texas, um, and honestly, I don't know. There's got to be some degree of statistician and programming and that kind of thing, but his big claim to fame is um, data visualization. So he wrote a really nice book um, called just fundamentals of data fundamentals of data visualization. So it's a really nice book, and it kind of describes all the different plots you can make, and not necessarily how to make them, but what are the fundamentals of presenting data in a nice, easy to digest manner. But that's his main claim. If you were to, you probably, whenever you need to install this, I think you have to do a install.packages cowplot. But I already had that installed, so I just need to call my library theme, my library cowplot. So what's nice about there is one link. One of these links is to making multi-pane graphs with cowplot, and it's not the only thing that cowplot can do. It's actually a pretty small fraction of what cowplot can do and all the functions that are within the cowplot package. But so we have these two different graphs. We have the box plot and the um, the density histogram, and we can present those side by side with CalPlot. And one of the functions, one of the easier functions to deal with is this is plot grid. So this plot grid functions allows you to put two or more graphs together into the same plot. So it's the help menu is fairly easy to understand. The link kind of describes how it can be used, but really it's just call your two ggplots. So I'm going to call it inbox and the inbox. What is the other one? Dense plot. And so if I just do this, if I just call my plot grid and these two graphs, now they'll be presented side by side to each other. Now it's not going to be very pretty, right? But um, it is an easy way to have two separate graphs put together into the same figure. So this isn't very nice. Everything gets kind of stretched out in a not very pleasing way. And so we can arrange these different plots. Right now, the default is horizontally, but we can also change them to vertically. So I'll do. So if I say in column, I only want one column, then this will be realigned to be on the vertical axis. Right, so now that looks a little bit better. Again, this is just presenting the same data in different ways, um, but that looks okay. Uh, these plots aren't exactly aligned, so like the x-axes aren't exactly aligned. So we can call an align part of this function. If I say v, it'll be aligned on the vertical axis. So it should get these gray areas uh, pretty much lined up. And so the nice thing about this cow plot is not having to do this by hand, right? So I spent the solid majority of uh, my graduate education building separate graphs, putting them all into Illustrator, and aligning everything by hand. And it can take forever to do. Um, but with CalPlot, I don't have to do that anymore. I spend considerably less time editing my graphs by hand, and I can make 95 to 98% of what I want my graph to look like just with R, ggplot, and CalPlot. Right? These three functions make life really, really easy. I'd say life. It makes graphing uh, very convenient. And it just keeps getting fancier. What we can do with R and graphing, I mean, it's getting very, very fancy, and it's getting very easy to work with. Um, so one thing I did want to mention with the last thing I'll throw in here. This is honestly the icing on the cake, because we can label our graph. So whenever we make a whole figure, there's multiple panes. There's multiple different graphs inside the same figure. I would want to label my graphs like A, B, C, D, E, F, something like that, right? If you looked at publications, you would probably see that multi-pane graphs labeled A, B, C, D, and in the figure legend, it's going to be a description of Fig 1A, Fig 2B, etc. And so CalPlot allows us to label stuff automatically. So I can call my labels part and just define it as make a vector of A and B. Something's not right. I need a comma. Okay, and so that will now, hopefully this will look okay. Sometimes it looks terrible. Just in this here, we can always make things look better than what's presented here. But now each pane is labeled with an A or a B to tell me if I want to make, again, it's just less things that I have to edit by hand in say Illustrator or a free version of Illustrator called Inkscape. Um, so I can make between R, ggplot, and cowplot, I can effectively make any graph that I want, and I can make it look however I want. And so one of the links I provided are just all the different ways that we can present the same data. So one thing I didn't mention over on Wednesday, and it was something I wanted to talk about, was this overall density plot. Let's see if I can find the raw, just that by itself. Hopefully, almost. There we go. Whatever. Whatever. Okay, so on my density plot, right? So these are my links from the ensemble, and that's from the transdecoder. And so one thing I just wanted to kind of mention is like this this giant peak that we see from our transdecoder ORs. It's very possible it's a combination of both an artifact of transcriptome assemblies and something that could be biologically relevant, right? So in our ensemble transcripts, we have full length transcripts. We have pretty much any transcript that's ever been observed in mice. But in our transcriptome, right? Remember there's alternative splicing. So the transcripts that we actually see in liver or kidney or whatever, um, very well, on average, may be shorter than what we actually observe from the genome. So it's very possible that these transcripts are shorter than average due to alternative splicing, right? It's every liver gene isn't the full transcript fully expressed for every single gene that's present. And also, we have to remember there are some artifacts in the transcriptome assembly, right? We see the same annotation, we see the same sequence, sometimes repeated several hundreds of times, and it's probably going to be roughly about the same length. Um, so that might be driving a little bit of artifact, maybe driving this as well. But either way, this could be. It'd have to be explored further. We'd have to filter this transdecoder output down a little bit. But again, sometimes we see biologically relevant um, phenomena out of our Trinity or our assembled data, and sometimes we don't. Well, if we wanted to try to get every transcript, if we wanted to try to build this kind of distribution from expression data, we would have to get almost every single tissue, sequence it all, and build either separate or concatenated um, assemblies that include everything from every single tissue, right? Because we're only, again, the liver only samples maybe half of all the genes that are actually expressed. So if we wanted to get every single transcript of every single gene, we would have to sequence almost every single tissue from an individual to build those transcripts. We didn't do it. Whenever we measured expression, we mapped our RNA-seq reads to a known genic library, but we can map those back to the genome itself. When we map those back to the genome itself, it can tell us where our intron and exon boundaries are. Because remember, whenever we have our mRNA, all the introns have cut out, and so whenever we map those reads back to the genome, we'll see reads mapped to discrete locations that correspond to exons and exons only. So we that's part of how we know where genes are in a genome is by mapping, getting tissues, taking that RNA out, and then mapping those reads back to the genome. Figure out where those genes are expressed from and what those genes are. It's part of genome annotation. Um, all right, the last thing that I wanted to mention, and almost all of this stuff today is just leftovers. The only kind of new thing is doing the cow plot to put neighboring graphs next to each other in the same figure, so I don't have to do it later by hand. Like I don't have to save each figure individually and then put it together. And so that's really relevant, especially mostly for those who plan to go on to grad school, because you will have to make graphs. And if you can kind of figure this out now, you're going to have a heads up, because a lot of your peers are going to be making crappy Excel graphs. And if you ever make an Excel graph and I know about it, you're going to get a very angry email from me, because Excel graphs are atrocious. They're terrible and they're hard to work with. Believe it or not, it takes a little bit of practice, but we make considerably better graphs using R and ggplot. That's something that I wish I had learned as a grad student. Um, last thing I wanted to demonstrate to you, and this I'm going to call in the iris data set. So this is just the flower stuff. It tells a couple. I'm gonna do a couple of box plots. We'll get this a little bit fancier. And before I lose my rewrite the whole code out every single time you want to graph it out. It's much easier to just save it into a script and then call it whenever and modify it as needed. But for like 5 minutes, I'm not going to do that. I'm just going to type into the terminal. I'm not going to save anything. But let's get steeple width by let's just do a quick box plot. Let's just do what I wanted to do first. So plot. I'll do a box plot just on the steeple length. Geom. I want that filled in. Okay. So the last one concept I didn't present, I meant to whenever we talked about box plot, um, is another function that we can call. It's called the geom jitter. Um, so this box plot tells us where what our data, how our data is distributed. But what geom jitter does is put every single data point on this graph as well. So whenever I have this additional function, now it doesn't just show me the distribution of these data points, it directly tells me where every single data point falls in this distribution. So for the data we work with, when we have um, our uh, our merged link, some of our data frames include like um, 68,000 points. It's really stupid to do a geom jitter. But if we have less than 200, less than 300, something that we can actually visualize, it's not bad. And I see this a little bit more commonly in the literature, to see the geom jitter, because it is kind of nice. That's kind of a nice little trick. I'm gonna hang on to that one. I'll save it as something like box. So let me make just for playing around with um, calplot one more time. Let's compare uh, sepal length and sepal width. So we'll just make that plot real quick. I think I got to color this in as something, so I'll color it by species. Geom. So this is just a dot plot, geom point. Okay. Yeah, so everything's colored in. So and one of these links that I have, I think it's perfect scatter plots, is what's really cool. Let's see if I can pull it up real quick. Um, is say, so this is more or less the same graph, sepal width and sepal length. So this is off of the iris data set. What's really cool about some of these graphing functions is to be able to have a histogram of the x and histogram of, or sorry, a histogram of the x and a histogram on the y on the same figure. And taking it another step further is actually having the geom densities for the X and Y axis. I think that looks awesome. I love seeing a figure like that because it really kind of demonstrates where all these points are at. I think it looks great. And so it's not that bad to actually draw it. And there's more than one way to do this. But right, so we can get so fancy with these graphs, just plotting from using the same data, presenting histograms of on the X and Y or density plots on the X and Y. There's really a lot of stuff we can do. So I'll save this real quick as just, so then let's get this another fancier. So plot grid, box point. So I'll just leave it as is. I'll leave it as default, see how that looks when everything's just on the horizontal axis. So that's not too terrible. Again, we can modify every single one of these graphs before we merge them together. I think I can align them horizontally. But either way, I think you kind of get the idea of how the power of plotting tools are, how powerful these tools are to make really pretty nice graphs. So I think I'm just going to call it here. Over the weekend, I'll ask you to draw some plots out using what we've learned this week. But next week is largely devoted to using R, not for plotting, but to actually run a specific analysis for us. But I'll call it here for today.